The first form of active transport we're going to look at is pumps. Active transport involves the movement of molecules against a concentration gradient. This time we're going uphill from lower to higher. This uses ATP and a carrier protein. If we're going to spend energy, we want to make sure we get the molecule we want. So this carrier protein makes sure we specifically get the required molecule. ATP is used to change the carrier shape. These protein carriers may move more than one substance at a time. Some are called antiporters. They move one substance in one direction and one substance in the other direction. Symporters can transport two substances simultaneously in the same direction. One of the most common pumps is the sodium-potassium pump. Both sodium ions and potassium ions do have leakage channels. That is, we do have a few channels that are open that allow sodium and potassium to move in and out of the cell along their concentration gradient. The sodium-potassium pump works as an antiporter. It pumps against the sodium and potassium concentration gradients. It helps us maintain a high potassium level intracellularly and a high sodium level extracellularly. This helps maintain what's called the electrochemical gradients that's essential for the functions of both muscle and nerve tissues. It also allows for cells to maintain fluid volume. As we keep these ions balanced, it keeps water balanced. Water is going to follow these ions. The sodium-potassium pump is an example of primary active transport, and it's also an antiporter. Here we see sodium from the cytoplasm binding to its transporter protein. ATP is split, and when that happens, it changes the shape of this molecule, pushing the sodiums out. But we also changed the shape and opened up these two areas that can hold potassium. So potassium pops in, and we're going to pump the potassium against its concentration gradient. There's a lot of potassium inside the cell and not much outside, so when we flip the channel back the other way, we push potassium into the cell. We go back to what we had before. We can pick up sodiums, pump them out, pick up potassiums, pump them in. Antiporter, one out, one in. This is a primary active transport. Symporters typically work by secondary active transport. We have a protein carrier molecule that can recognize both sodium and glucose. So when we open up the gate, sodium can go in its place, and we just happen to have also a seat for glucose. We're using energy to pump the sodium in one direction, and we just let the glucose come along for the ride. Because they're going in the same direction, this is a symporter. The other kind of active transport is vesicular transport. Some molecules are just too large to have a hole in the membrane big enough to let them through. So this vesicular transport is how we get large particles and big macromolecules across the membrane, and they'll be in little membranous sacs. ATP is required to produce the membranous sac and move the substances. Exocytosis is when we're moving stuff out of the cell, and endocytosis is when we're moving stuff into the cell. Transcytosis occurs if we pick something up on one side of the cell, move it across the cell, and let it out on the other side of the cell. We see this happen in the digestive system membranes. And vesicular trafficking is when we have vesicles that were moving around in the cell. They didn't come in from outside and they're not going outside. They're simply moving material from one place in the cell to another. Phagocytosis is one type of endocytosis. Here, pseudopods from the membrane move around the particle we want to take in, and we form this vesicle. This vesicle is referred to as a phagosome. Macrophages use this to take in bacteria. Some white blood cells also use this way. Pinocytosis is another form of endocytosis. The plasma membrane folds inward, and some of the fluid from the outside is captured inside this little vesicle that we've made. So we're taking in liquids. The cell uses this to help sample the environment and to take in dissolved materials. Nutrient absorption in the small intestine occurs this way. Phagocytosis is the equivalent of the cell eating something. Pinocytosis is the equivalent of the cell drinking something. A third type of endocytosis is receptor-mediated endocytosis. This allows for a specific molecule to be taken into the cell. We will have receptors on the surface of the cell. And whenever these receptors have their molecule attached to them, it will trigger this enfolding to make a vesicle. Cells use this in order to concentrate materials that may be in limited supply in the extracellular space, or to limit the amount of something they take in. Because notice, when we take in the material, we also take in the receptor. If there are no more receptors for that material on the surface, the cell can't take any more in. We use receptor-mediated endocytosis to take enzymes in, low-density lipoproteins, iron, and insulin. Iron can be toxic to the cell, so having a certain number of receptors on the cell limits how much iron comes into a cell. Once all the receptors are full and inside the cell, they won't go back to the surface until they need more iron. Unfortunately, receptors can also let certain bacteria and viruses and toxins into the cell. For receptor-mediated endocytosis to work, there must be proteins that coat the inside of the cell membrane. This marks the area for endocytosis. These proteins make kind of a net on the other side of the membrane, and if you've ever gone fishing and thrown out the net and then drawn it back in, you know that you close it up into a bag. Well, that's sort of how these proteins work, like a net that closes off a bag that pinches off a piece of the cell membrane. Clathrin-coated pits are the most common. These clathrin-coated pits provide a route for endocytosis and transcytosis. Caveolae are another kind of protein. These can be used to capture specific molecules, things like folic acid, unfortunately the tetanus toxin too, and this uses transcytosis as well. We know that caveolae is involved in cell signaling, but we don't know its exact function. And then codomer is a protein that coats vesicles that are going to be used in that vesicular trafficking, moving material around inside the cell. So not only do these proteins mark the area for endocytosis, but they sort of also address the vesicle to where it needs to go in the cell. Now here you see some different ways that vesicles are used in the cell. Here is endocytosis with a clathrin-coated vesicle. As it comes in, the proteins detach so they can go and be reused to net up another vesicle if we need to. This uncoated vesicle can now fuse with a lysosome, and we can digest the material in here and turn it into nutrition. This may be a case of transcytosis where the material was brought in, but it's going to go straight across the cell and come out on the other side. Or we may use some of this vesicle to go back up and make part of the plasma membrane. So there's a lot of different places that these vesicle pieces can go. Exocytosis is kind of the reverse of endocytosis. Usually if the cell has something it needs to secrete or get rid of, there is some sort of cell surface signal or change in the membrane voltage that signals where the vesicle is supposed to go. The substance is enclosed in this secretory vesicle, and there are these snare proteins that are at the spot in the membrane where they need to attach, and we have the snare proteins here. So as they attach, we can kind of open both of these up. We can let the material inside the vesicle out and yet maintain a continuous cell membrane so that we don't break anything. Hormones can be secreted in this manner. Neurotransmitters are released from the axon in this manner, and this is also how the cell can get rid of waste products. Here is a photomicrograph of exocytosis. So here's the cell membrane. Notice how it's continuous, and what was once the inside of the vesicle is going to become part of the exterior of the cell membrane. Membranes exhibit potential. We have what's called resting membrane potential. Now, potential energy is the ability to do work, and if we're at rest, work is not occurring. This membrane potential is produced because the membrane separates oppositely charged particles. And since electricity is the flow of charged particles, there's the potential for some voltage across the membrane, and this occurs in all cells. So we say cells are polarized. They have an inside and an outside that has different polarity. This electrical potential energy is only at the membrane. We have to have the same number of ions inside the cell and outside the cell. We have to have the same number of positives and negatives inside the cell and the same number of positives and negatives outside the cell. So this voltage is not the result of an inequality of ions, but rather an arrangement of the ions. So how do we generate a resting membrane potential? Cytoplasm has as its primary positive ion potassium and as its primary negative ion charged proteins. Interstitial fluid has as its positive ion sodium and as its negative ion chloride. So you can see we've got a chemical gradient. We don't have a lot of potassium outside the cell, so potassium would want to naturally go out, sodium would want to naturally come in, and negative particles tend to follow positive particles. We have to have equal positive and negative charges on both sides of the membrane. So we have that equality of charge, but we have different chemicals. The sodium and potassium pump continuously throws out 3 sodium ions from inside the cell and carries 2 potassium ions in. So we are constantly pumping sodium out and pumping potassium in. But we pump a little more sodium out than we pump potassium in. This is what gives us that chemical gradient, the fact that there's more sodiums outside and more potassiums inside. We do have sodium and potassium leakage channels in the membrane. These leakage channels would let potassium leak out and would let sodium leak in, but there aren't very many leakage channels, just a few. There are more potassium leakage channels, so more potassium leaks out. Now, as this potassium is leaking out, the negatively charged proteins try to follow it, but they can't cross the membrane. So they're sort of stuck on the underside of the membrane, sort of like that sibling that was left home with its nose pressed against the window. These negative proteins can't get out. Neuron cells and muscle cells can upset the resting membrane potential by opening gated sodium and potassium channels, and then we have something called an action potential. This does not happen very often, but it is a way for cells to send an electrical charge from one cell to another. So as we look at the processes that allow for a slightly positive charge on the outside of the membrane and a slightly negative charge on the inside of the membrane, we have potassium leaking out. And as it's leaking out, these amino acids try to follow it, so they get kind of pressed up against the back here. We have sodium being pumped out in greater quantities than potassium is being pumped in. Sodium is positively charged. And we have more potassium leakage channels, so we have more positive ions leaking out. That means that right here on the surface of the cell, we're going to have a collection of positive ions. There's a strong chemical gradient for potassium to try to get outside of the cell where there's not much potassium, and so that's why potassium is driven out of the cell. Those positive charges collect on the outside, the negative charges collect on the inside. Now, since potassium is the major player here, sodium plays a little bit of a role, but potassium is the major player in getting these positive charges out here. We say that potassium controls the resting membrane potential. Since the cell membrane is on the outside of the cell, it must interact with the environment. Cell adhesion molecules, or CAMs, help anchor cells to extracellular molecules, kind of like Velcro. They can help cells glide over each other, sort of like caterpillars crawl across the ground. CAMs can help signal if a cell has been injured or if it has a viral infection. Also, CAMs are sensors for making and breaking the adhesive junctions. And finally, CAMs can transmit intracellular signals. They can tell cells when they need to move to another location, when they need to divide, or when they need to specialize. The other interaction the cell membrane has with the environment is through its receptors. This is a group of integral proteins and glycoproteins. They're involved in contact signaling, the recognition of cells that are supposed to be in one place or another. They're also involved in the electrical signaling. They help with opening and closing the gated channels that are necessary for us to have muscle contraction and nerve impulse transmission. And they can act in chemical signals. Ligands, which are simply any molecule that can bind to a receptor, can bind to a receptor on the outside, and a message can be transmitted to inside the cell. In other words, the cell does not have to expend energy to take this ligand in, but its message can be transmitted to the inside. The cytoplasm is that part of the cell between the nucleus and the plasma membrane. There are 3 basic parts to the cytoplasm. The cytosol is the water and the nutrients. The organelles are the metabolic machinery of the cytoplasm, and inclusions are chemicals and stored nutrients. Only certain cells will have inclusions. For example, fat cells have lipid droplets. The liver cells will have glycogen granules. The mitochondria makes most of the ATP. It's involved in what we call cellular respiration. It is sometimes referred to as the powerhouse of the cell since ATP is our energy molecule. Mitochondria are interesting because they contain their own DNA, their own RNA, and their own ribosomes. They are sort of like little subcells. We believe at one time these may have been cells that were capable of living on their own. They were ingested by a larger cell, and this outer membrane is the phagosome membrane. In other words, this inner membrane is the cell itself, and the outer membrane is the phagosome membrane. Over the course of time, the mitochondria became dependent upon the bigger cell, and the bigger cell became dependent upon the mitochondria. Mitochondria can also divide on their own, independent of cell division. Cells that need lots of ATP will have many, many mitochondria. Liver cells and muscle cells, for example, have more mitochondria than other cells. Ribosomes are made of protein and ribosomal RNA, and typically they exist as two small subunits. They don't come together until they have to function. Ribosomes function to make proteins. This is the site of protein synthesis. Ribosomes can be free ribosomes, that is, they're floating freely in the cytoplasm, or they may be bound. They're attached to a membrane in the cell. The membrane they're attached to is the endoplasmic reticulum, or ER. The endoplasmic reticulum is this membrane labyrinth. It's a series of fluid-filled tubes and sacs that twist and coil throughout the cytoplasm of the cell. This is like the circulatory system of the cell. Material in these tubes can travel from one part of the cell to the other. Also, these membranes help separate the cytoplasm into individual spaces where certain chemical reactions can occur, sort of like separate laboratory rooms. Some of the reactions that occur in the cytoplasm would cancel each other out if they happen side by side, but if they're separated by a membrane, they don't know the other reaction is going on. We mentioned that some of the peripheral proteins on the cell membrane were enzymes because enzymes sometimes had to be attached to membranes. Well, the endoplasmic reticulum can act as a work table, another membrane where enzymes may attach while they're doing their work, and then they can detach and be free in the cytoplasm. Rough endoplasmic reticulum has the ribosomes bound to it. Smooth endoplasmic reticulum has nothing bound to it. Smooth endoplasmic reticulum is involved in lipid synthesis. The rough endoplasmic reticulum has the ribosomes attached. The proteins that are made by those ribosomes are very specialized proteins. They're folded into special three-dimensional shapes. Also, the rough endoplasmic reticulum can act as a membrane factory. Integral proteins can be made and placed on the membrane, and then the membrane can be taken to the cell surface and become part of the cell membrane. All of the proteins that are secreted from the cells are manufactured here, things like mucus. Cells that produce more proteins have more rough endoplasmic reticulum than other cells. The pancreas makes insulin, a protein. The liver makes a number of proteins involved in blood clotting and keeping the blood fluid. Plasma cells produce antibodies. These are also proteins. All of these cells would have a lot of rough endoplasmic reticulum. The smooth endoplasmic reticulum is involved in lipid metabolism, the manufacture of cholesterol and its breakdown. Because cholesterol is the basis for all of your steroid-based hormones, your sex hormones, we would see a lot of smooth endoplasmic reticulum in the testes and ovaries, since that's where these hormones are produced. Also, smooth endoplasmic reticulum, because it helps us break down cholesterol, are found in cells that absorb, synthesize, and transport fats. These particularly are in the intestinal cells. Some smooth endoplasmic reticulum helps break down stored glycogen to glucose in the liver, and a great many of our toxins and pesticides are lipid-based, and so the smooth endoplasmic reticulum is seen in liver cells and kidney cells that have the function of detoxifying drugs and pesticides. The Golgi apparatus is a series of flattened membranous sacs. This acts like a traffic director or a post office. It will modify proteins made by the rough endoplasmic reticulum and package them for secretion from the cell. So it receives proteins in transport vesicles from the rough ER, and then these proteins are modified in the Golgi apparatus, and then they're exported in secretory vesicles. Lysosomes are membranous bags containing digestive enzymes. These are used to rid cells of foreign substances and old cell structures. Lysosomes can merge with phagosomes and digest bacteria or viruses that may have been ingested. They can also merge with old cell parts, things that are worn out, and help digest those organelles. If the cell itself becomes sufficiently damaged, the lysosomes will rupture internally and the cell will be digested on its own. For this reason, lysosomes are sometimes called suicide sacs. Lysosomes are involved in breaking down glycogen to glucose and also in breaking down bone to release calcium into the blood. Peroxisomes are a lot like lysosomes. They're membrane-bound sacs that have powerful enzymes in them. These help detoxify harmful substances like alcohol and formaldehyde, and they help neutralize free radicals, which are byproducts of metabolism. The liver cells will have a lot of peroxisomes in them. All of these organelles we've just talked about, the endoplasmic reticulum, the Golgi, the lysosomes, and the peroxisomes, are part of what's referred to as the endomembrane system. So here you see the rough endoplasmic reticulum. The proteins that are manufactured will be given off in these secretory vesicles. Now these are going to be part of vesicular trafficking. They will go only from the rough ER to the Golgi apparatus, where those proteins will be put into the Golgi apparatus, and the Golgi will modify them. This is where some of our very complex proteins are made, not just plain proteins, but glycoproteins, lipoproteins, and things like that. Then once the Golgi apparatus is finished with them, there are three possible things that can happen. We can have a secretory vesicle. This will be tagged so it knows to go to the cell membrane, and the secretion will be released by exocytosis. We said that the Golgi apparatus was membrane factory. So proteins that are receptors can be popped up on this membrane and it can be pinched off. And here we have just replaced some of the membrane with this membrane and it will have the appropriate proteins in it. And then here is a vesicle pinching off that is actually going to be an organelle. This is going to be a lysosome. Those digestive enzymes are protein in nature, so they're manufactured in the Golgi. And then this one stays within the cell. Again, vesicular trafficking. This will be tagged to merge with a phagosome so that we can destroy or digest whatever is inside the phagosome and the cell can use it for energy. One of the homeostatic imbalances that can occur with cell membranes is a disease called Tay-Sachs disease. This is a genetic disorder that is seen mostly in Jews from Central Europe. Here, the lysosomes lack an enzyme. This enzyme will break down a glycolipid that's found in nerve cell membranes. Because the lysosomes can't break this down, the lysosomes take it in, but they swell with these undigested lipids, and this interferes with the functioning of the nerve cells. Infants with Tay-Sachs disease look like dolls. They have pink, translucent skin. Unfortunately, the symptoms of the disease are mental retardation, seizures, blindness, and typically these children die before the age of 5. The cytoskeleton is an internal framework seen in the cell. It helps support the organelles, transport them around in the cytoplasm, and is involved in cellular movement. There are 3 types of cytoskeletal fibers. Intermediate filaments are very tough. They form desmosomes. They help resist pulling forces. They're very tough, insoluble proteins. Microfilaments are more delicate. These are involved in cell motility. We'll talk about actin and myosin when we talk about muscle cells. These are the cells that have to contract. Microtubules are the largest of the 3 cytoskeletal elements. They're hollow tubes. They are involved in cell shape and cell division. So here we see the microfilaments, very thin. They can make this fiber network and the cell can change shape. The intermediate filaments look more like cables. They're very thick, twisted strands that allow for a lot of stability between cells. It helps keep cells connected together. And the microtubules are these little bead-like proteins that make this spiral shape, so this is a hollow tube. Now these can be very quickly lengthened or shortened, so these are involved in cell movement primarily. The centrosome is a structure that's located near the nucleus. It is sometimes referred to as the microtubule organization center. It contains the centrioles, which are two rod-shaped structures that are always parallel to each other that are themselves made of microtubules. The centrioles help develop mitotic spindles, and they're also involved in forming the cilia and the flagella. Cilia and flagella are organelles for motility. Now, both of them have this basic 9+2 tubular arrangement. That is 9 tubules in a circle around 2 central tubules. Here you see it there. Cilia are shorter and tend to cover the surface of a cell. We find these in the respiratory tract. And flagella are longer and fewer in number. Sperm have a flagella. A modification of the cell membrane is microvilli. These finger-like projections that help increase the surface area of the cell membrane. Microvilli are found on cells whose primary function is absorption. If the idea is that you're going to absorb material, the larger the surface area, the more likely you are to absorb material. We find this kind of cell lining the digestive system. We also find them in parts of the kidney. So here is our generalized cell again. We see the plasma membrane. We now see mitochondria, lysosomes, endoplasmic reticulum, Golgi apparatus. Here are secretory vesicles, and we have exocytosis going on. What we now need to look at is the nucleus.